it's my pleasure to introduce our next speaker for the afternoon, we have Jason Pell, who is a graduate student in the Computer Science Department at Michigan State University in East Lansing, working with Titus Brown. And he's going to talk about a problem that dovetails very nicely with the previous talk, which is the digital normalization of genome sequencing data.
Thanks, Fernando. I'm glad that Ziv gave a good introduction to some of the issues in next-generation sequencing. There's a little bit of overlap with what I'll be talking about as far as background, but the focus of this talk is mostly on handling metagenomic data, and I'll go into that later on as to what metagenomics actually is and how it's maybe not the best term since it's not really meta, but I'll get into that in a second. So our problem in the lab, at least one of the issues that we are trying to address, is how can we come up with good metagenomic assemblies? And this boils down to the classic assembly problem that has been around for the past couple decades. And as I mentioned before, we have an original DNA sequence, and then this gets shredded up by whatever sequencing platform, Illumina 454. These are some of the newer names, or PacBio and the USB thumb drive that was mentioned previously. But there's a bunch of different methods to come up with fragments, and next generation sequencing really results in error-prone short reads. And nowadays, Illumina produces maybe 100 base pair to 150 base pair reads. PacBio can produce much longer reads, but they're much more error-prone. So it's kind of a weird situation where the classic technology, which is called Sanger, it produces long reads about 1,000 base pairs, but it's actually much more accurate. The only issue is that it's much more expensive. So biologists are drawn to next generation sequencing because it's much more cost effective, and if you have a non-model organism, using next generation sequencing is in your budget, while Sanger sequencing is pretty much out of the question. So this is what a FASTA file looks like. If you have a DNA sample and you go to a sequencing facility, you might get a file back in FASTA format. You'd probably get quality information as well, and that'd be in FASTQ. There's other formats available, but this is a pretty simple one. You just have the name of the read, and then in this case, looks like that's about 60 base pairs. And you depending on how much sequence you generate, you can end up with millions of reads or billions of reads. And what we do in handling this type of data is we break up the read into k-mers, which was mentioned in the previous talk as well. And so this highlighted in red here is a 20-mer. So if we set k equal to 20, we go along the sliding window and take all of these 20-mers and then load them into, in this case, a count-min sketch, which was discussed in depth in the talk this morning. And we've been using a count-min sketch or a counting Bloom filter quite a lot for the past 2 or 3 years, and it's really enabled us to do a lot more than other labs have been able to do with the same amount of compute power. And so I'm just glad that it's still being discussed, and that other people are taking advantage of the data structure. So anyway, DNA assembly is very difficult, primarily because of base calling errors, as I was mentioning before, and those can also be caused by sequencing platforms. Each sequencing platform has a different error profile, and so Illumina, the ends of the reads are more error prone, and that's pretty much the case for all of the sequencing platforms, but each has its own technical difficulty that is difficult to generalize. And another problem is repeats. And that's especially a problem in mammalian genomes, especially human, where up to 50% of the genome is repetitive. And so if you're breaking up a repetitive region into k-mers, it's very difficult to unravel how big that repetitive region is when you have so many more errors, because if you have deep coverage of a repetitive area, you get tons more errors, and of course that will lead to a more complex assembly graph. And so it's pretty much the case that DNA sequence assemblers pretty much choke on repetitive regions, and it's very difficult to get a good assembly in repetitive regions. But a lot of recent sequencing efforts, like the human gut microbiome, human resequencing, and cow rumen, they've all needed 512 gigabyte machines to assemble their data sets. And the issue is that this is really not sustainable, given that the sequencing technologies are only producing more and more data. We really need to develop new algorithms to properly handle these data sets. Now, it's particularly difficult in metagenomics, and it's probably better termed ensemble sequencing, which is basically where you sequence an environmental sample like soil or just get a water sample from the sea, and you want to find out what microbes are in that sample. When you do that, there is a whole lot of different coverage levels between different species in the sample. So you could have a dominant type of microbe that you typically sequence more of than other microbes. But this kind of relates to the challenge of RNA-seq that was mentioned before, because you have some genes that are expressed more than others, and if you want to get all of the novelty in a sample, you really have to sample deeply. But then that relates back to the problem where some areas get covered more and they get more errors, and then you have a complex graph. So it's really like you can't win either way. And metagenomics is also very important because 99%, it's estimated that 99% of microbes cannot be cultured in a laboratory. So E. coli is one example of an exception to that rule, where you can put it on a petri dish and it'll grow, and you can sequence it, and you can poke it and do whatever and study it in detail. But the vast majority of microbes, we cannot do that kind of analysis, and sequencing it is right now the only way to access what's going on in a sample. So another side note is, I guess one of the interesting facts that I learned as a computer scientist learning biology is that for every one human cell in the body, there are 10 microbial cells. And given that 99% of microbes can't be cultured in a lab, we know quite a lot less about the human ecosystem than you might think. So in our case, what we are studying is soil metagenomes, and this is part of a collaboration with the Department of Energy Joint Genome Institute and Lawrence Berkeley National Lab, where we obtained soil metagenomes from Wisconsin, Iowa, and Kansas. The goal is to compare soil treatments. So, what's in prairie soil versus soil that's been farmed, or maybe a place that was restored to switchgrass or something like that. Essentially, we want to find out, how the microbes in soil change during different or in different treatments. So they generated a lot of data for us, and MetaHit on the top here is the human gut microbiome sequencing project, and that generated 578 gigabase pairs of data. So that's almost a petabase, or I guess a trillion base pairs of data. And In our case, there's this dark green here, and that's the latest sequencing that was given to us, and then the lighter green is, it's the same sample, but it's a slightly older Illumina platform. But you can see that they've given us tons of data and not a lot of computational resources to actually analyze it. And so rumen is another example where they took they got the metagenome from a cow rumen, and this was published in Science, and they required a 512 gigabyte machine. Same with MetaHit. And so we didn't really have the computational resources to handle this because soil is a whole lot more diverse than cow rumen or the human gut. So we ended up using a Countman sketch, and we really had to look at the data in a different way. So looking ahead, we realized that sequencing is becoming cheaper. People are only going to sequence more, and we can't just store the entire assembly graph in memory or just error correct and then load that in, or quality filter. You really have to look at it in a different way. And so a primary reason why I'm here is to try and think about next generation sequencing data as a streaming problem rather than a problem where we have the data set on disk, and we load that in and analyze it and leave it on the disk. Instead, we think that it's better to throw out memory or throw out data rather than hold on to it, because it's very easy to generate more. And it was mentioned previously that these algorithms should be the case where as n increases, you should get a better result. But in fact, some assemblers, when you throw all of the data at the assembler, you actually get a poorer assembly than if you just randomly select half of the reads. And so that's really just not acceptable, but it's really the reality, because that's just how the algorithms are, how they function. They expect a certain coverage level, and for metagenomics, or even just genomics, where there are a lot of errors and highly complex regions in the graph, that's just the way it is. And so this graph was shown in the previous talk as well. And at genomics conferences, this type of graph is always met with eye rolls and that kind of thing. But it is important just because it shows that it really doesn't make sense. It's not going to make sense to store this type of data on a hard disk when you can pretty much just regenerate the sample. And if the thumb drive sequencer is in vaporware, you can just have it get sequenced right off of your laptop. So I just want to cut back to soil. Soil is very diverse. There are millions of species, or microbiologists tend to call it operational taxonomic units, just because the definition of species is very, very fuzzy. But as you add the number of sequences, it really there's just tons more novelty. And with the data that we have, and this isn't really representative of that, but we really just always keep finding novelty the more that we sequence in soil. And even though the idea of coverage is a little bit strange in the context of metagenomics, we don't think that we've really reached like 1x coverage of some of our soil metagenome data sets. So what we have developed in this lab, in Titus Brown's lab, is a very simple idea. He calls it digital normalization. And essentially what we do is we go through every single read in the data set. As you recall back from earlier when I showed the FASTA file. And we basically check and see what the median kamer count is. So we go through and get the abundance level from the count-min sketch for each kamer, and then find the median. And if it's below some cutoff, then we'll actually update the counts, load the kamers into the count-min sketch, and then we keep the read, but otherwise we just throw it out. And that's essentially where the streaming idea comes from. And the idea here is that in soil or other environmental samples, we want to even out the coverage level between different species. So if you have a species A that is 10 times as abundant as a species B, the idea of digital normalization is that we throw out all of this unnecessary data and try and feed an assembler something more sane that just has a more even coverage level. And depending on if you're sequencing E. coli or some other genome or a mRNA-seq sample or a metagenome, you have very different distributions depending on what it is that you're sequencing. But once you apply digital normalization, it evens out the coverage level so that it's all the same in your reads data set. And so in this case, all of these three data sets were pulled together, and then digital normalization was applied to them all at once, and we get a similar distribution. Another advantage to digital normalization is that if you think about just the true 20mers in a data set like the E. coli genome, even if you sequence with 100x coverage, the E. coli genome, you still get some 20mers, true 20mers that didn't actually exist, or that should exist in the data set, but that just didn't get sampled properly. But when we apply digital normalization, we really don't lose that many true 20mers, but we also keep a very small number of the reads. And so essentially, when we try and assemble these data sets, we are using a very small portion of the reads, the original reads, but we're getting, in many cases, a higher quality assembly. And it also greatly reduces memory needs. So you can see that, depending on what the sample was, we can get anywhere from 22.4x improvement in memory to over 100x improvement. And a constant factor is maybe not important theoretically, but practically it's very important. So our soil metagenome samples, we went from not being able to assemble the data set to being able to do it in less than 256 gigabytes of memory. And these results maybe aren't that useful to the machine learning community, but we were able to assemble the Iowa corn and Iowa prairie data sets using digital normalization and another technique that we call partitioning. And we obtained about the size of the human genome in sequence, and also 5.3 million to 6.8 million protein coding gene regions. And the human genome has been estimated to have about 23,000 genes, so there's microbes are much more gene rich than mammalian genomes or other eukaryotes. So it really gives us a whole lot more information than if we were to just not use digital normalization and have a lot of the regions get thrown out because of very high coverage. So just to summarize, DNA sequencing data sets are becoming prohibitively large, and our solution, we believe, is to try and treat the data as a stream rather than just a big block of data that you have to analyze. And also, we've had great success with the Countman sketch data structure, and of course, the digital normalization algorithm that Titus and everyone else in the lab has worked on really does even out the coverage level and makes it a whole lot easier for assemblers to actually assemble these data sets. And our software is publicly available on GitHub if you're interested. It's under an open source license, and we're always trying to build upon it and improve it. So that's pretty much everything. This is the list of people who have been involved in digital normalization in our lab, the GED lab, and collaborators, Jim TG at MSU, and others, and there are our funding sources. So thanks.
So we have a few minutes of questions.
So one thing that I think people are really worried about in terms of this idea of streaming sequences rather than storing them, especially if you look at the medical applications, they are very afraid to throw away data that can later be used in a better way, basically, and then, in terms of even legal terms, right? I mean, you could have done this diagnosis, you have all the data, where's the data? Oh, we threw it into streaming. So, can you comment on this? I mean, what should we store, what shouldn't we, how can you decide?
I mean, I can't really give a better answer. I mean, certainly there are ethical issues to be worked out, but ultimately, I think it's an unresolved question. Yeah, I don't have much more to comment on other than, to me, it would depend on the data set, and certainly if we're thinking about clinical samples, then we would probably want to hold on to that, perhaps, but maybe basic science or something like that, then I think it's okay to throw away data. But yeah, it's certainly something to consider.
Actually, it's saying about being okay to throw away data. You publish a paper, and somebody wants to challenge your conclusions, and if you don't have the data to be able to back them up, that gets really in the way of reproducible research.
Yeah, and our lab's actually very big on reproducibility, and certainly if you're publishing a paper, you need the raw data to replicate your results. So I would say that if you're publishing a paper, you need to have the original data, like absolutely. So, yeah.
Any other questions? I actually have a quick question which dovetails into both of those, which is that since this is effectively a form of, I guess, speculatively lossy compression, do you have any understanding of the properties of the error that are made? In some compression schemes, say JPEG, for example, we know, not only do we know how does it compress an image, but we also know exactly what kinds of errors does an increased compression rate produce. So you can basically make an educated judgment with the JPEG image on how far you can go or what is going to be the impact of pushing harder on the compression, because we know the spectral properties of the algorithm very well. Do you have a sense and intuition or an understanding of what happens in terms of error with your approach?
Yeah, we've been looking into that, and currently one of the issues is that before, if you look at the contigs that an assembler produces, once you enter a highly complex region of the graph, then that's where the contig ends, just because the assembler chokes at that point. But with digital normalization, we've found that the contigs, we get longer contigs. It's just that the very ends of the contigs get cut off, and we're still diving in to try and understand where some of these errors are coming from. But yeah, it's something we're actively looking into. Is what happens when we are applying this type of lossy effect to the data.
Actually, just a quick comment on reproducibility to your question. As Jason mentioned, Titus's lab is big on it, and the paper that was published for digital normalization, which was made available at the archive preprint, was actually made available alongside with an Amazon AMI image that contains the data and the code ready to run. The full analysis takes about $4 worth of Amazon time. So basically, anyone in the world who wants to replicate exactly their analysis can boot up a virtual machine that has all the code that is ready to go, and you log into a web interface and click on an icon, and it re-executes the analysis, and then that is executable code. So you can then tweak it in the web interface, change parameters, and basically play with it. So on their website, they actually made it available, which I think is great. Okay, so let's thank our speaker again. And we have a
available, but this is a pretty simple one. You just have the name of the read, and then in this case, looks like that's about 60 base pairs. And you depending on how much sequence you generate, you can end up with millions of reads or billions of reads. And what we do in handling this type of data is we break up the read into k-mers, which was mentioned in the previous talk as well. And so this highlighted in red here is 20-mer. So if we set k equal to 20, we go along the sliding window and take all of these 20-mers and then load them into, in this.
It's my pleasure to introduce our next speaker for the afternoon, we have Jason Pell, who is a graduate student in the Computer Science Department at Michigan State University in East Lansing, working with Titus Brown. And he's going to talk about a problem that dovetails very nicely with the previous talk, which is the digital normalization of genome sequencing data.
Thanks, Fernando. I'm glad that Ziv gave a good introduction to some of the issues in next generation sequencing. There's a little bit of overlap with what I'll be talking about as far as background, but the focus of this talk is mostly on handling metagenomic data, and I'll go into that later on as to what metagenomics actually. The only issue is that it's much more expensive, so biologists are drawn to next generation sequencing because it's much more cost effective, and if you have a non-model organism, using next generation sequencing is in your budget, while Sanger sequencing is pretty much out of the question. So this is what a FASTA file looks like if you have a DNA sample and you go to a sequencing facility. You might get a file back in FASTA format. You'd probably get quality information as well, and that'd be in FASTQ. There's other formats actually is, and how it's maybe not the best term since it's not really meta, but I'll get into that in a second. So our problem in the lab, at least one of the issues that we are trying to address, is how can we come up with good metagenomic assemblies? And this boils down to the classic assembly problem that has been around for the past couple decades. And as I mentioned before, we have an original DNA sequence, and then this gets shredded up by whatever sequencing platform, Illumina 454. These are some of the newer names, or PacBio, and the USB thumb drive that was mentioned previously. But there's a bunch of different methods to come up with fragments, and next generation sequencing really results in error-prone short reads. And nowadays, Illumina produces maybe 100 base pair to 150 base pair reads. PacBio can produce much longer reads, but they're much more error-prone. So it's kind of a weird situation where the classic technology, which is called Sanger, is it produces long reads, about 1,000 base pairs, but it's actually much more accurate.